Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum and welcome back to our cell biology, genetics and evolution class. We were discussing genetic engineering in a very basic way. In fact, this particular series of videos is just an introduction to genetic engineering because there is a whole lot more to this subject, to this topic, which you will discuss inshallah in your final semester. Anyway, we were discussing the tools of genetic engineering, and in the previous video, we have seen that the major tools are restriction enzymes, the vectors, and DNA ligase. So, we are going to discuss first of all the restriction enzymes or the molecular scissors because they cut the DNA molecule at specific sites. So here it is. These enzymes, since they are known as scissors, and as you know, scissors, what they do, they are used for cutting something. So these enzymes play exactly the same role. They cut DNA, but not randomly, and this is what makes them a great tool, an indispensable tool, a very important tool in genetic engineering because they cut DNA at a very specific site. Now, where did these enzymes come from? These enzymes are actually found in bacterial cells like E. coli and other bacterial cells. And why they are found in bacterial systems? Because these enzymes, they are actually part of a very specific what we call RM system in bacteria, or restriction modification system, which is found in bacteria like E. coli and others. So they are actually part of that particular system, these restriction enzymes. So what they do, they restrict what the foreign DNA that enters in bacteria, like the DNA of a phage, a bacteriophage, and also they modify the host or the DNA of the bacterial cell so that it may not be attacked by its own restriction enzymes. Because if it can cut a DNA, it can cut a DNA of a phage as well as DNA of bacteria or bacterial cell, and which can be its own DNA. So to protect its own DNA, bacteria have a restriction and modification system. Obviously, this is not our topic and we are not going to discuss it now. And this is just to tell you that they are actually part of a system which functions in bacterial cell. So in this system, these restriction enzymes, they hydrolyze any exogenous DNA, not only exogenous, but endogenous or the DNA of the bacteria itself. So it can cut any DNA. However, the host DNA is modified so that they cannot act on the host DNA or its own DNA. Now these enzymes, actually these are groups of enzymes, and we have classified them into three types, and they are simply named as type 1, 2 and 3. So we have type 1 restriction enzymes, type 2 restriction enzymes, and type 3 restriction enzymes. Obviously all these three types, they play somewhat different roles as far as restriction or cutting of DNA molecule is concerned. Of these three, this is the most common type, the type 2 restriction enzymes. They are used very commonly in genetic engineering tasks. And now we consider very briefly the naming system, or how we name the restriction enzymes. So a common convention is that we name the restriction enzymes according to the bacterium from which we have isolated this particular enzyme, or the enzyme which is isolated from a bacterium. So it is named according to that particular bacterium. So for example, we have an enzymes which we have isolated from Escherichia coli. So this naming convention is that we take the first letter of the generic name, the genus name. So in this case E, and then we take the first two letters of the specific epithet. So it becomes ECO. So this is the name of the enzyme. Therefore, the first three letters, as I have written it here, they are taken from the name of the bacterium. E from the first letter of the genus and CO from the first two letters of the species name or the specific epithet. Now this RI, now this letter is actually a strain of E. coli, and this is actually one Roman one, first one. So the first one that was to be identified or purified. So the name of this particular enzyme EcoR1. So this is the genus letter, first letter, the first two letters of the species name. This is the strain of E. coli, and this is one, first one. So this is a naming convention or method of nomenclature. Another example is Bacillus amyloliquefaciens. Now in this case we have again B, the first letter of the genus, AM from the name of the species, first two letters of the species name or the specific epithet, and then we have this H, this is strain, and then again one, the first one, first that was identified from Bacillus amyloliquefaciens. So this is the naming convention, and we are going to see a few other names on the next slide. So this is how we name these restriction enzymes. Now, as far as the action of the restriction enzymes is concerned, so as this statement says, that their action is highly specific. They would act on a particular sequence of DNA, that is a particular sequence of bases in the DNA, for example, A, T, T, A, G. This is just an example or a fictitious example. So any one enzyme, say it is E, Co, or I, so it would act on a very particular sequence in the DNA and not other. So they are highly specific. And as it is just an elaboration, explanation of this sentence. So recognizes a specific sequence of bases in the DNA and it acts on that particular sequence or cuts it. Now, they are also known as endonucleases, as I have just told you in the previous video. We also saw it, endonuclease. The reason for naming them or calling them endonuclease, because this is a DNA molecule, so they would not act on the DNA from ends, that is from this end or from this end. They would cut the DNA from inside, that is inside the chain. That they can make a cut according to the sequence which the enzyme recognizes. So it will cut any of these parts. So since they cut the DNA from inside the strand, so they are known as endonucleases. Now let's take a look at some of the recognition sequences which are used by the restriction enzymes. So what is the length of the base sequence which is recognized by a particular enzymes? So here it is. So it can be a 5 bases sequence, 4, 5, 6. So this is a usual number, 4, 5, or 6. So some of the enzymes, they recognize a 4 base sequence, others 5 base sequence, and other 6 base sequence. The length of the sequence in which is recognized by these enzymes. So since they recognize different sequences of different length, so different restriction enzymes would create different fragments of DNA. For example, this is the DNA, and we have restriction enzymes, say A and B and C and D. So if we allow A to cut this particular DNA, it might cut it into only two parts. Say it is recognition sequence is here, so it would cut here. So the DNA, now DNA will consist of two fragments. B, for example, it has two recognition sequences, one here and one here. So it will cut the DNA from this point and this point, so we would get 1, 2, 3 fragments. Another enzyme like C and D, for example, they have 5 or 4 or 6 recognition sequences, so that will make a very large number of fragments depending upon the recognition sequences. So this is what it means, that the recognition restriction enzymes, since they have different recognition sequences, and then how many recognition sequences are present in the DNA on which a particular restriction enzyme is acting, we get fragments of different sizes, not of the same size. So this is what this particular statement means. So this is the explanation what I have already explained, that the size of the fragment, it is very closely linked to what the frequency of occurrence of the recognition sequence. How many times the recognition sequence is present in a particular molecule of DNA. The greater the number of sequences, higher will be or greater will be the number of DNA fragments. If there is, say, only one recognition sequence, so this DNA would be cut into two parts. And if the frequency is greater, for example, we have 6 recognition sequences, so what we get, one piece, second, third, fourth, fifth, and sixth. So we get 3, 4, 5, and 6 pieces. So since in this particular DNA and for a particular restriction enzyme, the number of recognition sequences is great, so we are getting a greater number of fragments as a result of the action of restriction enzyme. So this is a short table taken from the book Genetic Engineering, and it is displaying various enzymes. As you can see, we have just seen these two, BAM-HI and EcoRI. So these are their recognition sequences, and these small arrows tell you where they make their cut. So these are the places where they cut the DNA molecule. So we have EcoRI, HAE3, and this again 3 is that it was identified. This is the third enzyme identified. So HPA1, we have PST1, and then SAU3A, and so forth. So these are different enzymes, and as you can see, they have different recognition sequences, and the cut they make, because it is a double cut. If you cut, since DNA is a double-stranded molecule, if you cut only one strand, you haven't cut the DNA, it is still intact. So the cut may be made on both the strands. So as you can see, these cuts, they are known as staggered cuts. They create a sticky end like this. So as you can see, we have sticky ends. These are hanging overheads which help in the ligation. So these are various enzymes, these are their recognition sequences, and where do they make their cuts in that particular DNA sequence, recognition sequence, and what they create, what are the kinds of ends that they create. So obviously some of the things are beyond the scope of our discussion as far as this video or lectures are concerned, as regards the topic of genetic engineering. So these were different restriction enzymes. In the next video, we shall discuss the DNA ligases and more genetic engineering. So thanks for watching, and see you in the next video.